Welcome to today's session titled The Biology of Aging and Our Body's Readiness for COVID-19 Vaccines. Before we get started, I'd like to go over a few items so you know how to participate in today's event. When you joined today's webinar, you selected to either join by phone call or computer audio. And if for any reason you'd like to change your selection, you can do so by accessing your audio pane in your control panel. You will have the opportunity to submit text questions to today's presenters by typing your questions into the questions pane of your control panel. You may send these questions in at any time. We will collect these and address them during the Q&A session at the end of today's presentation. I would now like to introduce John Feather, CEO of Grantmakers and Aging. John?
Thank you, Kat, and welcome, and thank you for everyone who has joined us today. We have a large group of people today on a call, which indicates the tremendous interest in this topic. As Kat said, I am John Feather, the CEO of Grantmakers and Aging. We are so pleased to partner with the American Federation for Aging Research on this important webinar series, Aging and COVID-19, What Does Science Actually Tell Us? Today's webinar is the third in a series in which scientists and geriatricians will present the research behind care, lifestyle interventions, and programs that can help decrease older adults' vulnerability to COVID-19. Afar experts will share perspectives that can influence clinical, social, and self-care, and inspire new collaborations between scientific, medical, aging services, and philanthropic communities. We want to thank Afar for their partnership with this webinar series, and also thank the Research Center's Collaborative Network and Next Avenue for their promotional support. As mentioned earlier, you can enter your questions for our panelists at any time in the question box on the right side of your screen. We will collect these and read the questions during the Q&A portion. I'm now happy to introduce Dr. Stephen Elstead to introduce our topic today and our esteemed speakers. He is the co-principal investigator of the National Institute on Aging's Nathan Schock Centers of Excellence Coordinating Center. He is also a distinguished professor and department chair of the Department of Biology at the University of Alabama at Birmingham, and senior scientific director for Afar. Steve?
Thank you, John, and welcome everyone to today's session on COVID-19 and vaccinations. We have two distinguished speakers for you to talk to today. Dr. Janko Nikolic-Zugic is the department head and professor of immunobiology and the co-director of the Arizona Center on Aging of the College of Medicine at the University of Arizona Health Sciences. He's an internationally recognized immunologist and gerontologist, and I'm sure you're all going to be excited to hear his presentation. Our other speaker is Dr. Jenna Bartley. She's an assistant professor at the University of Connecticut Health Center on Aging. She studies translational research into human aging with a focus on immune responses and how physical function declines as we get older. So rather than take up any more of the time, I will turn things over to Dr. Nikolic-Zugic. Janko, please go ahead.
All right, now I'm finally unmuted. Thank you. And do I have the control of the slides, please? All right, so I will start talking, and then as soon as I get the control of the slides, I will move them. I will be discussing a couple of topics related to the virus and the disease that it generates and how it interacts with our immune system. The SARS coronavirus 2, which is causing this coronavirus, has a couple of unique things that it does to our body. I am not seeing that I can move the slides. Maybe you can move them for me for now until you give me control.
You have control, so click the mouse and arrow forward and backwards.
All right, great, thank you. Wonderful. So people who have done the work here are listed here. My colleagues from the lab up on top and our collaborators in the bottom, and they have been all wonderful in performing the research that I will be talking about. So let's talk a little bit about what is this virus, how is it different from other coronaviruses, what is the disease that it generates and what's unexpected in it, and then we'll turn the attention to our immune system, our immune response that deals with this virus in the adult and older populations, including the duration of immunity, and we'll talk a little bit about the basis of potential immune vulnerability in older adults, how this might relate to protection by vaccination. So this is the diagram of the virus, and you can see that it has on the outside these parts that are labeled with S or spike. This is the outermost protein that the virus has, and that is the first thing that is different between this virus and other similar coronaviruses. That is the part that has changed, that is, as we call it, mutated to gain access. So all viruses, viruses cannot multiply outside of our cells. They have to get inside the cell in order to multiply and make more copies of themselves. And so the first step they need to efficiently achieve is to enter our own cells. So this piece of the virus on the top of the spike protein that we called RBD, or receptor binding domain, is the one that has changed. It sticks to our own cells really, really well, much better than any other coronaviruses so far. And that is one of the keys to why this virus spreads so well. It is also relatively hardy, and it is made by our bodies at large numbers, and it's made in the upper respiratory system, where we can very easily cough or put it into the droplets as we speak loudly or sing or whatever, or shout, and that's how it spreads. And that is much different compared to its nearest cousins, SARS-1 or MERS, which are the viruses that are very, very deadly, but they do not spread nearly as well as this virus. It's also shed from the infected person, as you guys know already, about 2 to 3 days before anybody feels sick. It is a respiratory virus, which by definition makes it different for us to contain, because, as I said, we speak, we spread it out, we cough, we sneeze, we spread it out. All of these things contribute to its being very, very contagious. And it is also possible that it is being spread by people who have no symptoms at all, asymptomatic spread. And that's something that we're still studying, trying to understand how much is that really important. So the disease that the virus generates is a respiratory syndrome dominantly, and it has these very well-known symptoms of fever and cough and fatigue. But then it gets into shortness of breath, and in the most severe cases, acute respiratory distress syndrome and the cytokine storm. And the cytokine storm means that our immune system is trying to react to the virus, trying to kick it out, is not successful, and it ends up harming us more than it's helping at some point. That happens only in a very small subset of people, but it still is very, very dangerous and very, very, very deadly. So, as you're probably well aware, this is a disease that's very dangerous to older adults. And these are the stats here directly from the state of Arizona, but they're pretty much true across the world, that people between 45 and 54 years of age already have 3 times higher likelihood to die than those in the younger categories, 21 to 45. And then this goes up tenfold to in people 55 to 64, and up to 50-fold in people over 65. So really, really pronounced vulnerability to the disease. So we have an immune system that's exquisitely good at dealing with the virus, and we have some early sensor systems that alert us to the presence of the virus. But it is really our white blood cells, the T and B cells, our lymphocytes in the body, that eventually deal with the virus. And so we have 2 main classes, T cells and B cells. B cells make antibodies, T cells help B cells, and they also directly kill virus-infected cells. So T cells are really the main traffic controllers of our immune system. They're important both for directly dealing with the virus and coordinating the response overall. Our T cells are made in a gland that sits behind our chest bone and above our heart, and it's called the thymus. And this here is the diagram of the thymus, and it tells you that the thymus in young individuals is much bigger than in the old individuals. And that difference is actually already about a 10-fold difference in size by the time we reach puberty. The important part here is that, you know, cells that are made from the thymus are this sort of rainbow coalition of very, very diverse cells. They can defend us against any infection. They are in charge of defending us against new infections that we have not seen before. So the important point here is that when the thymus is making a lot of these cells, we're in a really good shape. We have a large pool of cells that are ready to defend us against new infection. When these cells get encounter their microbial pathogen like a virus, they will change their status. They will become memory cells, which means that they will be able to react much faster and better and stronger. And then this memory pool fills as we grow older. The problem is that the factory itself stops working. It doesn't stop working entirely, but it really reduces its output a lot early on in life. And so it drops by 10-fold by the time we hit puberty, drops by another 10-fold by the time we reach about the age of 50. So we're really down to about 1% of what we used to make, you know, by the time we reach 50. So we kind of like start running out of the players that are going to protect us against these new infections. And that is why new infections such as this one are a big problem for us. You know, when we make them, we actually then maintain them for very long periods of time. For decades, we keep them, you know, in our other lymphoid organs, like in our lymph nodes and in our spleen and so forth. But that also fails with aging at some point. So the story of our T cell generation is that the new T cell production diminishes early. Peripheral maintenance takes over, lasts for many decades. But really in the last third of life, we start running out of some of these cells. And we're down to roughly about only 1/3 of what we used to have in youth. So that is our first problem, our problem number one. Our the detection of the microbial pathogen happens the moment that the microbes enter. So let's say that these little red things here are a virus. Let's say that this virus came through the skin, or in the case of this respiratory virus, it would come through the epithelium of our mouth and our nose. It's immediately picked up by some very important cells called dendritic cells. They carry this virus in a very directed fashion to meet the T cells, and that all happens in our lymph nodes. So these cells come in, they flow into the lymph nodes. T cells come from the thymus and through the blood, and they exit in the lymph node, and this is where they wait for the dendritic cell to bring their own virus. We have billions and billions of T cells, and only a few of them are going to be specific for each virus. That is why we need the diversity. That is why we also need the numbers. And we kind of start running out of both when we grow older. So this dendritic cell comes in and is supposed to meet its own right T cell that is here. So why that is important. So this dendritic cell is the only cell that can activate the T cell. The T cell now will multiply many, many times. It will multiply. One cell will generate 100,000, if not 1,000,000 cells. And that is how we actually now have an army of cells that are going to attack the virus. So these cells here will come and help the B cells make antibodies. B cells are activated by the virus in a different manner themselves, but this interaction between T and B cells is important. And that is what's going to give us a long protective antibody response. What goes wrong with aging is, you know, several things. So first of all, you know, as I said, in the adult, this recognition of microbial pathogen of the virus by the dendritic cell works very well. Dendritic cells get big, juicy, and they talk to a lot of T cells, and we have a lot of T cells in the lymph nodes. And as a consequence of this interaction between these cells, we'll see this proliferation, and we will have a large, robust army of very well-armed T cells that are going to go out and zap the virus. The problem that we have with aging is that this initial detection is not very good. Dendritic cells don't pick up the virus very well, but even more importantly, perhaps, the number of T cells that they have to interact with is now greatly diminished because the thymus is not making them. And as a consequence of this interaction, we will not have as many defenders, and they will not be as well armed, and we will be a little delayed at coming to the battlefield with all of these cells. So this is really the problem that we're facing here. So what do we know about this virus? What did we do about this virus? So we wanted to study immunity to it. We wanted to figure out what parts of the virus are targeted by the immune system, and this spike protein, the part that has mutated that I told you, was one of the top targets. We wanted to measure the duration of the immunity and then figure out, is there an immunological basis to the vulnerability in older adults primarily? So what we did here is, like, in the beginning of March, we started to put together an antibody test to detect antibodies. And so this was the first interview that we gave for the papers. And I have said in that interview that we will use these antibodies just to sort of like track the virus. We weren't really planning to screen the population or anything. Fast forward about 2 weeks later, our governor announced that we are going to be testing about 250,000 people with our test. That was the same test that I was talking about. So we were in for a bit of a surprise. We had to scale up this test to test the population of Arizona. We generated a very highly accurate antibody test. And this test was a two-step test, making it the most accurate out there on the market. That allowed us to figure out and to drive down the possibility of wrong calls, particularly in terms of the false positives, down to very, very low numbers. We have tested roughly about 30,000 people so far. You can see here our preliminary data. Our seroprevalence was around 1% back in May, but is now more like 3 to 5%. And these are the data that we had from different populations. A lot has been made about it. This virus may not give us long-term immunity. There was a couple of articles that were really misinterpreted, saying that coronavirus immunity may be unachievable, that antibodies may disappear, and that we might lose them in a few months. That was all fortunately wrong, and it was wrong because our antibodies that we make are very, very long-lived, and these are the cases of other viral infections. You can see here that the half-life of antibodies for measles, for instance, is 370 years. And that's only the amount of time that you need to lose half of your antibodies, not to lose them all. So in some cases, that is much less. It's like 12 years for tetanus vaccination, but it's still very, very long. So it was very unlikely that we were going to lose antibodies very soon. And part of the wrong interpretation came from the fact that we ramp up production of antibodies very quickly, and then they go down because this is an emergency response by some very short-lived cells that are making antibodies. But as these cells disappear and therefore the antibodies start to decline, we make very, very long-lived responses that come and supplant them. And those very long responses last for months and years and decades. And that has happened, in fact, really with SARS coronavirus. And this is what we have found, that antibodies against different parts of the virus really last very long. So this is days post-onset. We've measured up to 7 months. Obviously, we have to wait to measure longer, but two different parts of virus, very long immunity. Most important, the antibodies that will neutralize the virus, that will prevent its entry inside the cells, also lasted very, very long and were very, very stable. So we weren't worried about this. This is all now published by now. We turned our attention from here as to what's happening with an older immune system. So this is an older individual that we have studied in a highly multidimensional manner to see which populations of cells may be changing. This is a severe case of SARS coronavirus, day 6 and day 13. And you see that this person in the beginning had a lot of antibody producing cells. These are the plasma cells here and the B cells down here, as well as a lot of T cells. So these are the CD4 cells and CD8 cells. You can see that by day 13, many of these cells were gone. So there's very powerful remodeling of these blood cells in this person, particularly the white blood cells. We're still trying to understand what's happening, but for the sake of time, I will just summarize here what I have shown you. Our conclusions are that correlation between immunity and disease severity in the literature right now appears stronger than those with age. We're not seeing too many older people that seem to be coming down with really low immunity to this virus. So a lot of more subtle things could be happening. The response may not be as timely. We could not have the right flavors of immunity, but just in terms of the magnitude of immune response and of antibody production, we're not seeing that old people are not making antibodies right now. That is confounded by some other things that we don't know. We don't know, for different people how much virus they got in the first place. So we need to study a lot of things. We saw this remodeling of immune cells in the blood with severe disease at some points. And then in people who recover, this seems to be coming back. We're starting to define signatures of innate and adaptive cells. And so the long and the short is that there's a lot here to study. But I'm fairly optimistic that we will have a good vaccine, and we will be discussing vaccines and vaccination through your questions and some additional comments, and through what Jenna also has to say. And so I will stop here and let her speak. Thank you.
Thank you, Dr. Nikolic. Now turn things over to Dr. Bartley to talk about exactly how vaccines get employed.
Great, thank you so much. Great, thanks. So Dr. Nikolic has reviewed very nicely how the immune system really declines with aging and why this is such a problem with COVID. This has been shown elegantly in mice about how the immune system declines as we get older, and we really see this clinically in older adults with COVID infection. So there are many studies in mice of ways to improve the immune system, but we don't always see this translated to clinical care. And I'm sure many of you guys have seen the headlines of cancer being cured in a mouse study or something similar to that, but yet most of them aren't actually translated to the clinic. And well, translational studies, that is connecting the findings from the laboratory, which is our animal studies and in vitro modeling and these preclinical models to actual human clinical care, has a lot of barriers. It's not easy. So going from this bench to the bedside has challenges. And if we look at the spectrum over here of translational research, from basic research, this is our animal models, all the way to translation to the community, there are multiple barriers. Here today, in terms of the body's readiness for a COVID-19 vaccine, there are multiple barriers in the early translational research with aging. So if we look right here, translating what we see in the lab to the actual people. So these preclinical studies that we do to develop vaccines generally test them in young animals, and our early translational studies are also in young adults. This is great from a safety standpoint and initial efficacy. However, it doesn't represent the responses that we'll likely see from vulnerable older adults. However, there's really no reason to despair because scientists have been working on different ways to improve vaccine responses in older adults for quite some time. So what do we know works in older adults to overcome age-related immune declines? Indeed, while COVID-19 is a novel virus, our experience trying to overcome the age immune system is not. We have great experiences from decades of research with other vaccines in older adults that we can learn from. For instance, the original shingles vaccine, Zostavax, was a live attenuated vaccine, but the newer Shingrix is a recombinant protein vaccine with an adjuvant. So an adjuvant is something that can be added to a vaccine to enhance immune responses. It can do this by a variety of ways, but overall adjuvants generally trigger sensors and pathways in immune cells to better potentiate the signal of danger. It basically tells the immune system to mount a more robust response. This adjuvant in the shingles vaccine is able to better stimulate the aged immune system and has a resounding efficacy over 90%. Similarly, if we look at the flu vaccine, we know that flu is still among the leading killers in older adults, but reduced vaccine responses leave older adults more vulnerable. Despite the variable efficacy of the flu vaccine year to year based on strain differences, the flu vaccine still reduces the severity of infection and the rate of hospitalization in older adults, even when the strains aren't an exact match that year. Importantly, to that end, there are 2 FDA-approved vaccines specifically for older adults. There is an adjuvanted flu vaccine, which uses squalene, which is actually made from shark liver oil and creates an oil emulsion that increases the immunogenicity of the vaccine. And then there's a high-dose vaccine, which actually just quadruples the hemagglutinin, so the dose of the antigen that you're priming the immune system against. So both of these result in improved antibody titers post-vaccination in older adults. So we already know, based on these FDA-approved vaccines, that there are specific ways to overcome the aged immune system, both by adding adjuvants and increasing the dose. These provide extra stimulation for the aged immune system to overcome the negative effects we see with aging that Dr. Zulich already went over. But the problem with this is that while they improve the immunogenicity of the vaccines, they require integration in the vaccine development, and they may be different for different pathogens, and all these require extra time and testing. However, in a more recent paradigm shift, research has questioned that rather than improving specific immunogenicity of a specific vaccine, what if we could actually improve aging and improve the aged immune responses themselves? And this is where I get particularly excited. So rather than targeting specific deficits in the aged immune system, geroscience approaches target the biological drivers of aging. These may improve clinical outcomes for a variety of age-related diseases, including immune responses against pathogens and vaccine responses. Thus, this geroscience approach is particularly attractive not only for this current pandemic, but in general to combat a variety of age-related diseases. These approaches target the hallmark of aging, which include stem cell exhaustion, altered intracellular communication, genomic instability, telomere attrition, epigenetic alteration, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, and cellular senescence. So these are a lot of different things that go on with aging, and they all impact how the immune system responds. But what's more important than each one of these individual hallmarks of aging is that in preclinical models, if we target some of these drivers of aging, they're particularly promising in improving not only lifespan, but health span as well. And most importantly, to the COVID-19 vaccine, key translational studies in humans have also been promising. So in particular importance for older adults' response to the COVID vaccine, there's been a few studies that have used this geroscience approach for vaccine responses in aging already. For instance, 6 weeks of mTOR inhibition with an experimental drug. mTOR is one of the key modulators of metabolism in the body, and it's been seen to kind of go a bit haywire with aging to impact immune responses. So if we inhibit mTOR with these specific drugs, what we can see here is that there's an increased antibody titers at 4 weeks post-flu vaccination. And then furthermore, if you voting to really this geroscience approach, what they also saw was that a year following treatment, so these people were only on this inhibitor for 6 weeks, for a full year following treatment, there's a reduction in infection rates among these individuals. So this suggests that the drug was actually able to rejuvenate, so to say, the aged immune response. Similarly, there are other geroscience-guided trials that are ongoing that examine the effect of metformin on vaccine responses in older adults. Metformin is a drug of choice for the Targeting Aging with Metformin or TAME trial that you may or may not have heard about. But this trial really aims to first test the geroscience hypothesis in this first-of-a-kind study where it examines if targeting the biological drivers of aging, you can actually reduce a variety of age-related diseases. And metformin is particularly attractive for this and for immune modulation because it does target multiple drivers of aging, and it also happens to have an excellent safety profile for over decades of use. So metformin is currently under investigation to improve the responses of the Prevnar pneumococcal vaccine for older adults. And more recently, I actually myself completed a pilot study earlier this year, right before the COVID shutdown, to examine the impact of metformin and flu vaccine responses in non-diabetic healthy adults. This translational study was based on some preclinical studies that I saw in mice where I was able to see some altered T-cell metabolism with aging. And since we know that metformin can modulate metabolism, it seemed like a good target. Additionally, in other mouse studies, metformin has been shown to improve T-cell memory responses in young mice, and diabetics on metformin tend to have stronger flu vaccine responses. So it seems like it might be a promising drug to really improve immune responses and vaccine responses specifically in older adults. Unfortunately, my lab was shut down for a while right after for COVID, so I don't have the full results yet. However, I do want to note that my study was more of a feasibility pilot study, so I don't have adequate power to really make definitive conclusions. I would like to note, though, that metformin was pretty well tolerated, and there was very few adverse events throughout the study. I'm hoping to have the full results by the end of this year and determine if metformin might be a promising intervention that we'd be able to use for not only flu vaccine, but also COVID vaccine when it becomes available. But importantly, the real promising part of these completed and ongoing studies is that these interventions are not specific to a certain vaccine, but they're rather using this geroscience approach to target the biology of aging to improve overall immune responses. Thus, these approaches represent a geroscience adjuvant, so to say, or a geroadjuvant to boost vaccine responses, suggesting that these geroadjuvants can improve overall immune responses and vaccination to novel pathogens in older adults. Additionally, along these lines, it's also worth mentioning that other geroscience-guided trials are currently underway, examining the impact of these different drugs on COVID-19 infection and prevention. These trials include metformin, other mTOR inhibitors such as rapamycin, as well as senolytics. And much of this research is underway now that will hopefully provide a good picture if this might be a way to really target the aged immune response, both in terms of COVID-19 infection, prevention, and vaccination. So what does this mean overall for the COVID-19 vaccine and the aged immune response? Well, even though we know that the aged immune system will likely not respond as well as a young immune system to the vaccine, it's still likely that we'll have some level of protection in older adults from the vaccine. And these geroscience approaches are particularly promising. And what's very promising about it is that they would be easily integrated into COVID vaccination because they wouldn't require integration in the vaccine development itself, but rather augmenting the actual timeline by augmenting the actual timeline of where you would be able to get the vaccine, have some sort of geroscience pre-treatment and get the vaccine to improve your responses. In the meantime, the whole world is waiting for the COVID-19 vaccine to become available. Really the best thing that we have to protect our older adults right now is for everyone to keep up masking, washing hands, and social distancing. And importantly, now more than ever, it's important to get your flu vaccine. It's likely that the COVID vaccine, similar to the flu vaccine, is going to be a situation where the more people we can get vaccinated, the more people will be protected, and the more vulnerable older adults will be protected if we can get as many people vaccinated. And that is all.
Thank you. Well, we have a number of questions coming in, and I think we'll just start going through them. And maybe, Dr. Nikolic, you can start with this one, and then we'll see if Dr. Bartley has something to add. Vaccinations often have side effects, and we have a question here about the development of autoimmune diseases associated with vaccination. And is there possibly another route to destroy the disease, perhaps from some cancer molecule? I'm not sure what this questioner is responding to, but what about autoimmune diseases because of vaccinations, Dr. Nikolic?
Sure. So, and as Dr. Bartley was also mentioning, there is this inverse profile of our reaction. The strong vaccines in general cause a lot of inflammation, and they cause long-lasting immune responses. So there's a trade-off between potential side effects and the duration and the potency of immunity. We have gone quite a long way in terms of understanding how to really unravel and split these parts into very well-defined signals. She has mentioned the contrast between the Shingrix vaccine, which is a live attenuated vaccine, although it's an odd one in that in terms of its profile, and the the Shingrix vaccine and the old Zostavax vaccine, which was live attenuated. So now we can actually really pick pieces of the virus, or maybe a single piece of the virus, add 1 or 2 molecules that are going to juice up our immune system to some extent, and have the really relatively well controlled reaction. So that is the way for us to go forward and to diminish the potential to cause unwanted reactions and unwanted autoimmunity. Part of the problem with autoimmunity is that people who have autoimmune disorders are already predisposed for very strong immune activation. And so, there is part of the answer is also in improving the screening. We generally tend to not screen our vaccinees before we vaccinate to figure out who would have potential problems and who would not. And I hope that modern medicine will move in that direction. We'll talk a lot about personalized medicine, which would be the medicine that's tailored to the particular person. And the more we can do that, the more we can do those quick screens going forward, the better off we will be. I'm not really aware that we could use cancer molecules or anything that's used in cancer therapy right now in these particular cases. In the case of cancer, the task is slightly different. We're trying to actually get our immune system to work even more strongly, because it has a problem of dealing with our own self tissues. And normally our immune system is programmed to be tolerant to our own self tissues to avoid the autoimmunity. So we're kind of like trying to get it to be a little bit autoimmune to destroy our own cancer. So I would leave that part for now until we actually convene the panel that deals with cancer, immunity, and aging, which I think is going to be a fascinating topic for the future.
Thank you. Dr. Bartley, someone asked that you mentioned several times in your talk a geroscience approach. What is geroscience, and what exactly did you mean by that approach?
Yeah, so geroscience is kind of a more recent, well, not so recent, paradigm shift where rather than correcting for individual things that go wrong with aging, it seems like it's pretty established that there's biological drivers of aging. This is seen in mice, in our flies, and all other different models, all the way up to humans. And what happens is if we could target these biological drivers of aging, we think that we can really slow down aging and prevent a variety of age-related diseases. So the TAME trial that I also spoke of will be the first of its kind trial to really test that hypothesis out in humans. But it has been very elegantly shown in mice that using this geroscience approach, so targeting aging, is able to improve not only lifespan in mice and other models, but also health span, and really, I hate to use the word rejuvenate, but is able to improve the aged responses in general.
Thank you. Dr. Nikolic, here's a question about people who presumptively had COVID-19, but they can't find antibodies in them. Is this the result of ineffective testing, or is something else going on?
Well, there was a lot of ineffective testing early on. I think that now we're on more solid ground in terms of testing. The cases where people have verifiably been infected and do not have antibodies are very, very, very rare, as are the cases of reinfection that has been mentioned over and over, but is in fact exceptionally, exceptionally rare. There's been a really great study on both types of responses in the Gulf, in Oman, where more than 130,000 people were verified positive. They had very strong antibodies. About less than 0.001% of the people actually had some evidence of reinfection. And that correlates roughly with about the number of people that might have defects of immunity, innate defects of immunity or other defects of immunity that we did not recognize before. So really having the virus and not having antibodies will be super rare. Having the virus and being reinfected is also going to be super rare. Those are going to be very prominent exceptions to the rule that more than 99.9% of the people that get exposed to the virus will have lasting immunity and will not get reinfected, at least within many months that we have verified and measured, and most likely several years.
Okay, thank you. A kind of a follow-up on that is no vaccinations are 100% effective. And someone has asked, if you get vaccinated, is there a way to test how well you've responded to that vaccination? Dr. Bartley, would you like to take that one?
Yeah, so Dr. Nikolic has actually mentioned, so we're able to actually look at antibody responses in these people. So the antibody levels in the blood will give you an indication of the response to the vaccine. And even though they do wane over time, as Dr. Nikolic very nicely showed, they peak and then kind of level out, you'll still be able to detect those levels. So there would be able to do antibody testing to look at efficacy of the vaccine.
Okay, great. Dr. Nikolic, says cancer chemotherapy treatments, including removal of lymph nodes, affect the body's ability to fight this virus or the response to the vaccine for that matter?
It does. The removal of the lymph nodes, fortunately, is less of an issue because we have 450 lymph nodes in the body, and so removing let's say 10 or 20 of them, which is a typical removal that would occur, for instance, in breast cancer, where you would take them out of the armpit, still leaves you with plenty of other lymph nodes where you can mount your immune response. Chemotherapy is a different animal that kind of like is there to systematically try to deal with cancer, and it is toxic to our immune system. So there's definitely going to be a level of immunosuppression, and this is one of the important topics that we still need to study to understand to what extent our cancer patient population, particularly, again, when you talk about aging in cancer, cancer is one of the typical diseases that increases with aging, as Dr. Bartley has mentioned, the geroscience approach. It's the one where, more than 8% of the cases happen in people over 50. So you've got a combined effects of both aging of the immune system and the therapies that can be toxic to the immune system. It is a very important population that we're trying to study and protect with all of these, really good geroscience approaches. But we don't have any answers right now along those lines.
All right, thank you. Dr. Bartley, we've heard of these people that are the long haulers that can't seem to shake all the symptoms in a timely fashion. Is there any evidence that older individuals are more likely to become long haulers than younger individuals?
It does seem clinically that we know that there's obviously a higher mortality rate in older adults, and it does seem like the infection itself tends to linger and have more kind of effects, and that probably goes back to all the deficits we see with the immune system with aging. So some of the inflammation that the virus causes and the damage to the lungs isn't as easily repaired in an older individual as it is with a younger individual. So I think that's probably the reason why we see these kind of lingering effects within our older adults that do survive the virus.
Thank you. Dr. Nikolic, do we know anything yet about people who are infected with HIV and how they are responding to this virus?
I really haven't, seen good studies that are dealing with this, but I'm sure that this is being studied as we speak. People with HIV have overall, even when the HIV virus is well controlled, a whole slew of symptoms that appear as accelerated aging very much. Some people have calculated in fairly elegant studies that having HIV seems to add about 10 to 15 years of your biological age. Now, that is an oversimplification, but it is also not inaccurate to say that you see a lot of symptoms of what you would normally assign to wear and tear in people with HIV much, much earlier. Also, their immune system has not quite healed from the HIV assault. It has kind of caught up, but it still bears some very prominent scars. So I would be very surprised if people with HIV did not have more severe issues with this particular virus.
Okay, thank you. So here's a question about I guess the current vaccines that are being tested. There are a couple that seem to be ahead of the rest, at least in terms of their effectiveness. Dr. Bartley, are these live vaccines, and what does it matter if they're live vaccines or not live vaccines?
Yeah, so there's a few different approaches to vaccine development, and Dr. Nikolic actually has touched on some of the more advances that we have now compared to before. So it seemed like we normally used to go with a live attenuated vaccine where we would somehow make the virus weaker so that it couldn't cause infection but would mount a robust immune response to it to provide protection. But now our technology is really just much better. So rather than doing these live attenuated viruses, sometimes we're able to use recombinant proteins. So we are really able to target the proteins that are needed to have adequate antibody production to cause neutralizing protection. And we're able to use different adjuvants that stimulate the immune system or different approaches like that. So to my knowledge, I don't think anyone is working on a live COVID vaccine for the front runners. I'm not sure about some of the other ones in earlier stages, but it seems like really our technology now is just where we can have much more exact precision approaches to vaccines, that these recombinant proteins with different adjuvants and different things of that nature, RNA vaccines, really are able to stimulate the immune system better and are safe and effective.
I'd like to add just one little thing to that one. I also am not aware of anyone trying to make an attenuated vaccine based on a coronavirus. There's a couple of platforms that are using recombinant adenoviruses, which are different types of viruses, and they're inserting pieces of the coronavirus. I am completely with Dr. Bartley in this one. I think that our front runners, we have plenty of ones that are really using recombinant proteins in different delivery platforms that are showing exceptional efficacy, really good antibody production, like, again, not endorsing anyone, but like Novavax, for instance, has a really good platform. GlaxoSmithKline with Sanofi have one platform. There's a couple of mRNA platforms that seem to be causing a little more side effects. So we have to wait and see, but I'm confident that we will have good, safe ones that are efficient and that are recombinant proteins in different delivery systems.
Okay, thank you. So here's a question. I forget which one of you mentioned vaccines could be expected to have, quote unquote, some level of protection. And one of the listeners says, that doesn't sound particularly encouraging. What exactly did you mean by some level of protection?
Yeah. So I think what I meant is in older adults. So we know that vaccine responses in general are diminished with aging. So if you were to look side by side, a younger individual to an aged individual, the same exact vaccine, it's pretty likely that the older individual is going to have a reduced antibody response and protection from that vaccine. But that doesn't mean the older individual isn't going to have any protection. They're still going to mount some immune response and have some level of protection. It just likely isn't going to be as robust as the younger, healthy individual. So I didn't mean to cause any despair. Older adults will still be protected. I just think that the same level of protection won't be seen in an older adult compared to a young, healthy individual.
So I just like to put that in a slightly broader context. People need to understand that actually no vaccine has really been solely designed for older adults ever. What the two examples that Dr. Bartley mentioned is that we quadruple the amount of the dose in the flu vaccine. Is kind of like almost the most sophisticated thing that we have ever done. We have now an added adjuvant, which is good, but it's been so long coming to make better vaccines specifically for older adults. And we do have one reason for optimism, and that is that the Shingrix vaccine, the one that has replaced the live attenuated one, is actually 97% efficacious in older adults. So we have one very big, bright, shining light, and we have a lot of more knowledge that we can deploy in that way. So I'm optimistic, but we need to be cautious about this one overall as well.
Thank you. This is a question for Dr. Bartley about metformin. If it turns out to be a useful pre-treatment, is this something that would be taken once or a few times, or something that would be taken over an extended time period?
I mean, that's really hard to say. So, like at this level, my guess would be that it would take kind of a few weeks before vaccination to really improve the inflammation in the older individuals and kind of reset so that they're able to respond to the vaccine better. But it really hasn't been shown clearly in any, even in animal models yet. I mean, it's all based on theory. I hopefully will have some results from my study very soon to see if that time frame. So I had my patients were on metformin for about 10 weeks before vaccination. So we'll see if that might work. Since metformin has relatively low side effects, I don't think a longer pre-treatment would technically be bad. But it obviously takes some optimization and some precision to it, because I don't think it will be as easy as we give you a couple tablets of metformin, you get the vaccine, and now you're going to be great. You know, there's a lot of science that goes into that, the optimal dosing and really optimization of that precision.
Thank you. So Dr. Nikolic, someone would like you to speak to the cytokine storm, what that is, and is it more common in older people, younger people? Does age not matter?
That's another great question. So, you know, cytokine storm is the word that describes the fact that our immune system goes into overdrive under certain circumstances. And we know that there are some bugs that are really good at, you know, making the cytokine storm happen directly. Like, you know, you'd go and eat some ice cream, you know, that has some, you know, streptococcus, you know, I mean, staphylococcal and enterotoxins and stuff like that. It'll make you violently ill within hours. You know, you'll have all sorts of aches and pains and diarrhea. That's sort of like a typical acute cytokine storm. The cytokine storm that occurs as a consequence of this particular virus, SARS-CoV-2, is not very well understood. It doesn't happen in the majority of people that have severe SARS coronavirus disease, COVID-19. It happens in a relative minority of the people, even with the severe disease, and why it happens, as I said, we don't know. A common thinking would be that we haven't been able to kick out the virus sufficiently in those people, and the immune system is going like, oh my God, what am I going to do? I'm going to ramp up, and I'm going to make. So cytokines are the hormones of the immune system that are short acting, and they're supposed to be short lived, and you're supposed to make them, you know, in a sort of like, you know, high spike, and then you turn them off. But if you have prolonged generation of cytokines, they're going to make you very, very sick. And that is really what happens with the storm. And again, it's very likely that it is the combination of somebody's particular genotype and the propensity to react to certain stimulation and of this virus that generates it in a fraction of people. But how and why it happens in those people and not in others, we really still don't understand. I do not think that it is more likely to happen in older adults actually than in others. But again, that's something that still needs to be distilled and studied.
Okay, we have a lot more questions, but I think we only have time for a few more. Let me ask Dr. Bartley this one. There's been a suggestion, I'm not sure where, I haven't seen this suggestion, that flu vaccine might actually make people more susceptible to COVID. What would you have to say to that?
I actually haven't heard that one. Everyone hates on the flu vaccine for some reason. I don't understand it. No, I mean, I can't think of any logical scientific reason of why that would happen. The flu vaccine is well designed. It targets only the flu virus. There's many studies that shown having flu vaccination has reduced risk for not only just getting flu and reducing hospitalization, severity of infection, etc., but also has some off targets of reduced risk for stroke and a few other cardiovascular events. So I find it pretty hard to believe that it would have any increased risk for COVID. I didn't see that one though.
I haven't either. Dr. Nikolic, do you have anything to add about this?
No, I mean, I would completely just second what has been said. I haven't seen it either, and there's no basis in science or medicine why that would in any way, shape, or form impact adversely the coronavirus. In fact, I would say, like, the only thing that would be the side effect of taking the flu shot is not having flu while you might be having COVID, which I think would be a good thing overall. So.
Okay, well, I think we just have time for one more. So let me put this to both of you, and maybe Dr. Bartley, you could answer first, and then to Dr. Nikolic. Are there any things that we can do to sort of boost our immune response? You're reading all the time about taking this supplement, this vitamin, eating these foods. So are there any suggestions you could have to people to try to keep their immune response as strong as possible?
Yeah, I think, unfortunately, I wish I could say, buy this supplement, do this, do that, and you'll be protected. But most of those supplements are not really scientifically backed. The best things to do to boost your immune system are, exercise. I believe there's a webinar tomorrow, actually, that will go into more detail about that, how the importance of exercise for keeping your immune system fit and able to respond to pathogens. And then just generally eating healthy, getting a good amount of sleep. Just the general things that keep you healthy is really the only things that have been shown to really help here. Unfortunately, there's no supplement that's going to be a cure-all or prevention.
Dr. Nikolic, anything to add?
I think this is my favorite time of any Q&A. I mean, this question always comes whenever we have and we always come up with same old, same old. I mean, you have to keep yourself healthy, by sleeping, eating, and exercising well. There are supplements that you need when you grow older, because you're not able to absorb enough of them. But finding them in a good bioavailable form is really important. I mean, that is a billions and billions dollar industry. I don't know whether it has reached trillions yet. That is completely unregulated, as we all know. And so people peddle things, take a lot of this, and you look at it, that you can't even absorb it into your body. So, having the deficiency of zinc, for instance, needs to be corrected. Having enough D3 vitamin is important, but you need to talk to your doctor and find the one that is good and can be actually used by your body, rather than popping pills randomly and buying them, at Walmart in the form that you can never absorb. So those are really, really important points.
All right. Well, thanks to you both, and thanks to everyone who's sent in questions. I'm sorry we didn't have time to get to all of them, but I'd like to now turn things back over to John Feather.
Well, thank you so much. Really, really informative and useful information for all of us in this often confusing and certainly anxiety-producing time that we're all living in. So I want to thank Dr. Stephen Alstead, Dr. Jenna Bartley, and Dr. Janko Nikolic for being with us today and sharing their incredible insights and wisdom. We also want to thank our partner, the American Federation for Aging Research, and the Research Center's Collaborative Network and Next Avenue for their promotional support. We will be making today's presentation available to anyone who wishes to view it later, including both the slides and the audio. Watch for an email to be sent out within a few days. And finally, I want to thank all of you for participating in today's webinar. Please stay safe.